Identification of clones by nucleic acid hybridization. Now, when you have a genomic DNA library or you have a cDNA library, there are multiple clones present, and out of which, if you were to identify a single clone that contains the gene of interest, the question that arises is how will you identify that single clone? So, therefore, nucleic acid hybridization is a way by which one can identify a clone of interest. Therefore, let us look at the learning outcomes of this session. Hybridization is a phenomenon that can be used as a tool to identify a single clone, as hybridization is complementary and sequence based. The identification of clones in a genomic library or cDNA library can be done by using a probe that can complementarily bind to only the gene of interest. Therefore, the probe has to be designed accordingly. Colony or plaque hybridization is generally used to identify a single clone, either by using a radioactive labeled probe or a non-radioactive labeled probe. For probing, it is important to have the probe as well as the target gene in a single-stranded form. Only when they are single-stranded can they complementarily bind to each other. So this is an important factor that has to be addressed when one is carrying out hybridization. So what do we understand by nucleic acid hybridization? When you have two single-stranded DNA or one single-stranded DNA and one single-stranded RNA or two single-stranded RNA, and each of these single-stranded is say from a different source, there is an ability of the single strands to base pair with each other if the bases are complementary to each other. And so that is what is understood by hybridization. So if there is or if there are two strands that are not homologous, when you say not homologous, it means that a very low percentage of bases are identical or complementary. And so there is some complementary, because there is some complementarity, there will be interaction or there will be base pairing of the two strands, but then the hybrid that is formed will be not very stable, it will be unstable. Yet there is hybridization happening. When will you have a stable hybrid? When you have a high percentage of complementary base pairs. That means more complementarity between the two strands, more is the feasibility of forming a stable hybrid. So you can see if the strands are homologous, then you are able to see highly stable hybrids being formed. One can also have, as mentioned earlier, hybridization happening between DNA and its complementary RNA. So therefore, these are the different ways or these are the different possibilities of hybridization, and each one of this is talking about the fact that there has to be bases that are complementary to each other. That is something that is highly essential. So what is colony and plaque hybridization? Now why do you need to have colony and plaque hybridization? Because when you take genomic DNA library, you will have all the clones in master plates in the form of colonies. If you're using bacteriophages as vectors, then you would have in a lawn of bacteria certain regions where you have more clear zones, and those are where you have plaques. So the bacteriophages, because they lyse the cells, you know that wherever there is bacteriophage, there there will be what is called as plaques. So therefore, uh, if you have 100 colonies in a master plate, out of the 100 colonies, it is important or you need to identify which is the colony that has the gene of your interest. And that is what is called as colony hybridization or the process of finding out that clone with that colony which has the clone of your interest is colony hybridization or plaque hybridization. Say for example, this is a master plate containing library of clones. For ease, let us consider that there are only two colonies that are there. Now, what is done as a step one in colony hybridization is that the nitrocellulose or a nylon membrane is pressed on the surface of the plate, because of which what is going to happen is that the bacterial colony, some bacteria will adhere to the nitrocellulose or the nylon membrane. So this is basically looking at the fact that whatever is present in the plate is now present in the nitrocellulose membrane or the nylon membrane. Now this membrane is treated with alkali and proteases because this bacteria has its cell wall, has its cell membrane, but what are you eventually interested in is the DNA. So therefore, the bacteria is treated in such a way that its cell wall is lysed, its cell membrane is lysed, and you have proteins being removed and therefore proteases being attached. So with this, what is going to happen therefore is every other part of the bacteria is removed except the DNA, which is still remaining bound to the nitrocellulose or the nylon membrane. Now, this DNA also on the nylon membrane is attached but not very strong interaction. It is not a very strong attachment. So if you wash with buffer, what is going to happen is that the DNA will also leave the membrane. So therefore it has to be ensured that the DNA gets immobilized on the nitrocellulose or the nylon membrane. So how is that done? So if supposingly one uses nitrocellulose membrane, then one can bake it at 80 degrees centigrade for 2 hours, because of which what is going to happen is that the DNA will cross-link with the nitrocellulose membrane or the nylon membrane through its sugar phosphate backbone. So the sugar phosphate will interact with the positively charged nitrocellulose or nylon membrane. So both nitrocellulose and nylon membrane are positively charged. And we know that sugar phosphate backbone is negatively charged. So you will have the negative and the positive charges come close to each other. And because of the baking now, this is going to be in a strong interaction, and therefore that is what is called as cross-link. After the cross-linking or after the immobilization of the DNA on the sugar phosphate backbone and its treatment with alkali, ensuring that the DNA is also denatured to leave only the single strand. So therefore what you see exposed over here is the nucleotides, and these nucleotides can pair with another single stranded DNA if there is complementarity. So a probe that has complementarity to the gene that you are interested in. So the probe that you should have complementary base pairs in such a way that it goes and binds only to the gene of your interest. So suppose this nylon membrane is now put in a solution that contains the uh, probe, and the probe has to be labeled. So it can either be a radioactive label or it can be a non-radioactive label. So if there is complementarity, then the probe will bind to the DNA that is attached or immobilized on the nylon membrane. Now this is washed thoroughly with buffers so that excess of the probe is removed. Any probe that is bound to the nitrocellulose membrane without any specificity, non-specific interaction, then it should be removed. What should only remain bound is that probe that has complementarily bound to the DNA of interest. So as you can see on washing, those probes that were not bound with high specificity are washed away, while only that probe remains bound to the nitrocellulose membrane because it has formed a complementary base pairs. Now one needs to detect which or where the probe is, because wherever the probe is, there the gene of your interest is. So what is done if the probe is labeled with a radioactive molecule? So then one applies on it an X-ray film, and then the X-ray film is exposed. So wherever you have the probe, there you would have a spot formed on the X-ray film, and that is how one can detect the probe. And when you're detecting the probe, you're indirectly detecting the gene of your interest. So on an X-ray film, what you observe is that this is where the spot is formed. So out of the 4 colonies that are say present in the Petri plate, okay, now why do I say Petri plate? Because this nylon membrane is actually a replica of what is there on the surface of the plate. So this nitrocellulose membrane, which had 4 bacterial colonies attached, out of the 4 bacterial colony, only this colony has bound to the probe, and therefore this is what is called as positive hybridization, and this has detected the gene of interest or the clone that contains the fragment of DNA that you are interested in. So this is what is colony or plaque hybridization. Now, labeling of probes is an important part of the entire entire technique. So the question is, how is the probe labeled? And if the probe is labeled, then what radioactive molecule should one use? So we know that nucleotides is made up of the sugar, the base, and the phosphate moiety. So what is generally done is that the phosphorus is radiolabeled. So one uses, instead of normal phosphorus, which is P30, one uses P32. And the it is ascertained that at least the alpha phosphate is labeled, because this alpha phosphate is going to be in a phosphodiester bond with the next nucleotide. So thereby, when you have several nucleotides present, the phosphates of each of these in the phosphodiester bond is labeled. So the labeled nucleotides can be incorporated into the probes, but how? So one of the ways by which the labeling is done, it is by a method that is called as nick translation. So nicks are gaps that are present in the DNA molecule. So as you can see over here, wherever there is a nick, you would have a 3' OH that is free, and that 3' OH can be used by DNA pol 1 to synthesize or to fill the gap. So therefore, for filling the gap, one of the nucleotides that is used is labeled. So for example, here ATP, DATP is labeled. So wherever there are complementarity for A, there the ATP which is labeled will go and bind, and you would have this entire probe getting labeled. Okay, so this is labeling by nick translation. Similarly, you can have labeling by end filling. In this case, what is observed is that the ends of the probe can be sticky ends. Say, for example, in this case, the ends are eco R1 sticky ends. So what can be done again in this case is that a lienofragment is a DNA polymerase 1 without the exonuclease domain. It only has the polymerase domain. So using the 3' OH and a labeled ATP, you can have the ends being synthesized. And so that is why it is called as end filling. But because the ends are filled, now you have the radioactive label at the ends. So therefore this probe is labeled again. Another method is what is called as random priming. So when you have the probe synthesized and it is a double-stranded DNA, you can denature it by heating to get a single strand. Now, to this single strand, you basically subject it to binding with random primers. So when you say random primers, primers that have very different sequences, but one or two of the primers may come and find complementarity on the single strand and bind. So here you can see, out of the many primers that are present, two of the primers have come and bound to the single stranded DNA. Now these primers with its 3' OH end can be continued to be synthesized by the lienofragment using a labeled nucleotide. So again, what you find over here is, as the lienofragment begins synthesis of the new strand of DNA, the new strand of DNA will contain the labeled ATP wherever there is a T. So if you have a T over here, you know that an A will come and bind over here, but that A is labeled. So therefore this entire probe will again get labeled because of the incorporation of a labeled adenosine monophosphate. Okay, that is the general idea. But this was all looking at use of radioactive label, but you can also label with non-radioactive components. So for example, one can have labeling of the probe which is non-radioactive. So for example, this is by adding a biotinylated nucleotide, that is incorporation of a biotinylated nucleotide. So you can see that you have a DNA probe to which can be added a biotinylated DUTP. Again, incorporation of the biotinylated DUTP can be done by nick translation, end filling, or random priming. But eventually what you get is a probe that has biotin attached to it wherever there is a U incorporated. And you know that U will always hydrogen bond with A. So wherever there is an A in the single strand of the DNA, there you would have U getting incorporated. And so now this probe is biotinylated. When you allow this biotinylated probe to bind to an immobilized DNA on the nylon membrane, it will go and bind wherever there is complementarity. It will not go and bind to all DNA, only to that DNA which has complementary base pairs. Now, after washing, when one has to detect where the biotinylated probe has gone and bound, what is added is avidin, and this avidin molecule is attached to several fluorescent markers. So, if the avidin goes and binds to the biotin, because avidin-biotin interaction is highly specific, wherever there is biotinylated probe, there the avidin is going to go and bind, and under the fluorescent microscope or under UV, one can observe where in the nitrocellulose membrane the fluorescence is happening, and that is the region, or that is the colony, or that is the place where you have the gene of your interest. Now similarly, one can also do labeling with horseradish peroxidase. So in this, a single-stranded DNA probe is taken, and then by using glutaraldehyde or other chemicals, the horseradish peroxidase is attached to the single-stranded probe. Now, when this single-stranded probe hybridizes with the complementary DNA on the nitrocellulose membrane, its substrate, chromogen substrate, is added. Peroxidase being an enzyme, it will break its substrate, which is generally H2O2. And during the chemical reaction, what can happen is a chromogen substrate luminol, which is added, can be acted on to give rise to what is called as chemiluminescence. So a glow of light is formed, and this glow of light can be observed. So wherever in the nitrocellulose membrane you have the glow of light, you know that that is the colony, or that was the place where the colony existed and had the gene of your interest. So this is about how probes are essentially important for hybridization, colony hybridization, and plaque hybridization. So let us make the conclusions. To identify a single clone from multiple clones in a genomic DNA library or a cDNA library, the concept of nucleic acid hybridization is one of the strategies used. In colony or plaque hybridization, the colonies or plaques are transferred to a nitrocellulose or nylon membrane, which is then treated such that single-stranded DNA remains bound to the membrane. A labeled probe specific for the clone-containing gene of interest is allowed to hybridize to the DNA on the nitrocellulose membrane. Autoradiography reveals the position where the labeled probe binds. And autoradiography is used only when you have the probe being labeled with a radioactive substance. If it is with a fluorescent marker, accordingly it is detected under UV, or if it is chemiluminescence, you just have to see the glow under light. And that is the colony plaque that contains the gene of interest. Wherever there is a glow, wherever there is a spot formed, in the X-ray, that is the colony or plaque that contains the gene of interest. So nucleic hybridization is the basis of using colony hybridization and plaque hybridization to identify a single clone containing the gene of interest. Designing the probe holds the success to identification. Thank you.